In this time, I'd like to talk about the identification of the acupuncture mechanism on brain reward circuit modulation. Everyone well knows that the acupuncture is a component of complex therapeutic and healthcare system of China that has been used for more than 2,500 years. In 1997, NIH said that acupuncture can lead to certain conditions, including drug abuse, stroke, rehabilitation, and headache. But recent acupuncture research has mainly focused on acupuncture stimulation site and target disease. So research on the mechanisms that deliver acupuncture's therapeutic effect is largely unexplored. For over 10 years, our research team has been studied about the acupuncture effect on the modulation of the mesolimbic dopamine system on drug addictive model. And we recognize the ascending pathway contributing to acupuncture stimulation transmitted from the peripheral acupoint to brain. To know that, we developed the mechanical acupuncture instrument and established the drug addictive animal model. And we'll analyze the various brain area activity during or after of the acupuncture stimulation. Based on our previous study, in this presentation, I'd like to talk about the three major topics. The first topic is which one is involved in the action of acupuncture in the peripheral acupoint? The sensory nerve versus the connective tissue. The second topic is identify the ascending spinal pathway contributing to acupuncture stimulation induced inhibition of drug-induced behavior. So in here, we recognize the two main somatosensory pathways in the spinal cord. And the last topic is the analysis of the various brain regions involved in the regulation of the brain reward circuits through the acupuncture stimulation. And here we recognize the medial prefrontal cortex, lateral habenula, and rostral middle tegment area, and mesolimbic dopamine pathway. So the first topic is which one is involved in the action of acupuncture in the peripheral acupoint. This research published in the 2019 in the Frontiers in Neuroscience under the title Peripheral Sensory Nerve Tissue but Not Connective Tissue is Involved in the Action of Acupuncture. Many studies suggest the initiation of the acupuncture signal, but there are two leading models of the initiation of the acupuncture signals, the neurologic model and the connective tissue model. In the neurologic model means the acupuncture signals being initiated by the action of sensory nerve ending and specific nerve fiber are transmitted to the brain through nervous pathway, resulting in a very tight psychological effect. And the connective tissue model means the twisting or rotation of the acupuncture needle created a connective tissue around the needle, and the formation of the collagen fiber, which initiated the mechanical signal transmitted the whole body network. So in this study, we want to know explore whether acupuncture effects are mediated through nerve or connective tissue. So results show that first we developed the twisting acupuncture instrument to mimic the clinical treatment of the acupuncture in human. So we control the needle movement, the speed or angle. So we choose the optimal effective condition and use the other experiment. So in here, we tested the acupuncture effect in the three different disorder model. This model is the acute cocaine-induced hyperactivity model. So when we treated the cocaine, The last will increase the movement. It means that the hyperactivity in the locomotion. When we treated the HD7 and acupuncture can significantly decrease the movement compared to the control. And we treated the acupuncture, uh, give the some bupivacaine and collagenase injection of the acupoint. The bupivacaine used to block the sensory nerve, and the collagenase treated to disrupt the collagen fiber. When we treated acupuncture, when we treated the bupivacaine, is uh, wholly block the acupuncture effect, but when they treated the collagenase, it remained the acupuncture effect. And this model is the stress-induced hypertension. When we treated acupuncture, we significantly decreased the blood pressure, but in here also, when we treated the bupivacaine, it's blocked the acupuncture effect. But collagenase treatment did not show the blocked acupuncture effect. It means the remain the acupuncture treatment effect. And last model is the colitis animal model. In here, when we treated the acupuncture stimulation, it significantly decreased the pain response compared to control. But in here, also when we treated the collagenase, it remained the acupuncture effect, but when we treated the bupivacaine, it's blocked the acupuncture effect. And next, we analyzed the morphological change after the collagenase injection at the acupoint. It showed that the wholly disrupted the collagenase, but the somatosensory nerve is remained. It means that did not damage after the injection of the collagenase. So this result suggests that the effect of acupuncture required the mediation by the nerve tissue, but not connective tissue. And the next topic is the identification of ascending spinal pathway contributing to acupuncture stimulation induced inhibition of drug-induced behavior. This result published in the 2017 in the scientific report under the title Spinal Pathways Involved in the Somatosensory Inhibition of the Psychomotor Actions of Cocaine. So there are two main somatosensory pathways in the spinal cord that relay the sensory signal from the peripheral to brain. The one is the dorsal-column pathway. This pathway transmitted noxious tactile information. The other is the spinal thalamic tract. This tract conveys the noxious signal, including pain and temperature. So in this study, we want to know to identify the ascending spinal pathway contributing to peripheral stimulation induced inhibition of drug-induced behavior. So first results show that when we give some surgical transection of the dorsal column pathway and cunate nucleus, and we treated the acupuncture stimulation. So results show that when we treated some surgical injection of the dorsal column pathway, is abolished the acupuncture effect on the cocaine-induced locomotor activity. And we treated and we recorded the neuronal activity by the extracellular coding. This result show that when we treated during treated acupuncture stimulation, significantly increased the neuronal activity of the dorsal column pathway. And we next the transaction of the surgical transaction of this spinothalamic tract. And this result show that when we treated the section, the acupuncture significantly decreased the locomotor activity. It means that this pathway is not involved in the acupuncture signal transmission. And next, we recognize the ventral posterior lateral nucleus, nucleus, and we give this ibotenic acid to give some chemical damage of this region. And the results show that when we treated the acupuncture stimulation, was, uh, the acupuncture effect was blocked after the lesion of the VPL site. So in this result show that the dorsal column pathway is involved in the uh, mediated the acupuncture stimulation, inhibition of the cocaine induced locomotion. And next, we recognize the lateral habenula, the brain side. So we uh, detected some temperature change of this area. Temperature is the one index of the recognized the neuronal activity. When we treat it during the acupuncture stimulation, that this area is significantly increased the temperature. And C-force expression is also increased after the acupuncture stimulation in the lateral habenula. And this neuronal innervated the RMTG, the BTA, and when we give the chemical region of the lateral habenula, and next we treated acupuncture, in this result show that acupuncture effect was not shown that after the lesion of the lateral habenula. And also we detected the neuronal activity change during of the acupuncture stimulation. So results show that when we treated the acupuncture stimulation, it significantly increased the neuronal activity of the lateral habenula. And when we give some lesion of the VPR, the acupuncture uh, increased the neural activity of lateral habenula, but after the VPR lesion does not show the increase of the lateral habenula uh, after the acupuncture stimulation. And next, we blocked the dorsal column pathway and somatospinal thalamic tract and treated the acupuncture, and then we recognized the moderation of the uh, nucleus accumbens activity. So results show that treated the cocaine was significantly increased the neuronal activity of the nucleus accumbens, and the acupuncture stimulation was decreased that. And dorsal column pathway, the blocked, it also increased the neuronal activity, although the acupuncture stimulation, but STT lesion is remain the acupuncture effect. So this result show that the acupuncture can moderate the mesolimbic dopamine system and the circuit, and those are column pathways involved in this signal transmission. And this result show that the lateral habenula is also involved in the acupuncture stimulation effect. So our present study show that stimulation of the 87, the acupuncture, suppressed the cocaine-induced hyperlocomotion, and the effect were completely abolished by the lesion of dorsal column pathway and lateral habenula, but not to the spinothalamic tract. And the last topic is the analysis of the various brain regions involved in the regulation of the brain reward circuit through acupuncture stimulation. Medial prefrontal cortex, lateral habenula, and RMTG and mesolimbic dopamine pathway. So this result show that this result published in the 2021 in the Addiction Biology under the title Acupuncture Reduced Cocaine Psychomotor Response by Activating the Rostral Medial Tegmental Nucleus. The rostral medial tegmental nucleus, the RMTG, has recently been considered as an important player in modulation of the mesolimbic dopamine system. This site is composed of GABAergic cells that send the front projection to the VTA synapse into the dopaminergic cells in the VTA. So in this study, we want to know the acupuncture if acupuncture at age 7 would activate the RMTG and reduce drug-induced NH dopamine and psychomotor activity. The first results show that when we treated the acupuncture with the cocaine, significantly decreased the locomotor activity compared to the only control a cocaine injected group. And yes, this blue graph will show the acupuncture effect, and the Li5 is the control site of the HT7. And when we treated the electrical lesion of the RMTG area, the results show that after the lesion, the acupuncture effect not show that. So it means that the RMTG uh, ability, the inhibitory effect of the acupuncture on acute cocaine-induced locomotor activity. And next, we uh, blocked the RMTG area by the optogenetic inhibition. So in this study, show that when we give some the hyaluronopsin in the RMTG area, when we treated the yellow light in here, so specifically blocked the RMTG area. The results show that when we during the light on with the HT7 does not show the effect of the acupuncture stimulation. So this results show that RMTG area is mediated the acupuncture effect on cocaine-induced locomotor activity. And this result show that it's confirmed the neuronal activity of the RMTG area during the light on and off. So when you give the light on, show that after light on with the HT7 stimulation, does not activate the RMTG area. And when we want to know the isolated neuronal type of the acupuncture activated neuronal type by the acupuncture stimulation. So we double labeled the immunohistochemistry with the GAD67 with the C4. GAD67 is the marker of the GABA neurons. So this result show that when we treated the acupuncture stimulation, was significantly increased the C4 and GAD67 merged cell. This result means the acupuncture stimulation significantly increased the GABA neurons activity. And the extracellular coding of the GABAergic neuron also increased the neuronal firing rate during the acupuncture stimulation. And next, we treated the PEPA, is agonist of the glutamate, so chemically increased the RMTH area without the acupuncture to mimic the acupuncture effect. So when we increase the neuronal activity by the chemical, and also significantly decreased cocaine-induced hyperlocomotor activity. This treatment also increased the GABAergic neuronal activity by the PEPA injection. And next, we explore the dopamine release change of the acupuncture stimulation in the nucleus accumbens by the fast scan voltammetry. So results show that when we treated the cocaine, was largely increased the dopamine release, but when we treated the acupuncture stimulation, it significantly decreased the dopamine release of the nucleus accumbens. And after the RMTH area lesion, the acupuncture effect does not shown. So this result means that acupuncture can decrease the dopamine release of the mesolimbic dopamine system, and in here the RMTG area is mediated, transmitted acupuncture signal. And this result is the lesion of the lateral habenula and fasciculus lateral flexus. This circuit is the RMTG area, neuronal intervention with the lateral habenula to the RMTG area. This area is also is mediated the acupuncture effect. And this result published in the addiction biology in 2023 under the title Mediation of Medial Prefrontal Cortex to Lateral Habenula Pathway is Acupuncture Inhibition of Cocaine Psychomotor Activity. In this study, we want to know the acupuncture at HD7 of the activated medial prefrontal cortex to lateral habenula pathway and thus reduce drug-induced psychomotor activity. So first result was more confirmed acupuncture effect on the cocaine-induced locomotor activity. So result show that when we treated acupuncture stimulation was significantly decreased the neuronal cocaine-induced locomotor activity, and the chemical lesion of the medial prefrontal cortex, the perilimbic side, results show that after the lesion of this site is blocked the acupuncture effect. And we recorded the neuronal activity of the perilimbic side will show that when we treated during the HD7 stimulation will show significantly increased the neuronal firing rate of the medial prefrontal cortex. So these results show that medial prefrontal cortex is mediated acupuncture effect, and during the acupuncture stimulation, this site was significantly increased the neuronal activity. And next, we confirmed the neuronal intervention of the medial prefrontal cortex to the lateral habenula. So we injected the associated adenovirus injection into the prefrontal cortex is anterograde to move the lateral habenula. So this figure shows that this neuronal intervention of this medial prefrontal cortex and lateral habenula circuit. And want to note the isolation of the neuronal type when we did the optopatch experiment in here. So when we treated the light stimulation, was significantly increased the EPSC of in the lateral habenula. And we treated the AP5 and CNQX, the glutamate receptor antagonist. As this EPSC is significantly blocked. So this result means that this neuronal type is maybe the glutamatergic neuron. This results show that when we give some halorhodopsin injection and we treated the yellow light to block this neuronal circuit. And after the blocked this circuit was significantly blocked the acupuncture effect. And this result also show that this prefrontal cortex is a mediated acupuncture effect on the dopamine release on the drug addictive model. Anyway, so this result show that acupuncture recruit the medial prefrontal cortex lateral habenula RMTG circuit to suppress cocaine-induced psychomotor response. So summarize our summarize my previous study, show that in the peripheral acupoint, when we treat the acupuncture stimulation, is activated the ulnar of A fiber, and this signal is transmitted the dorsal column pathway and the cunate nucleus in the spinal cord. And this signal also transmitted to the in the brain, so prefrontal cortex and this glutamatergic neuron was transmitted lateral habenula and RMTG, and finally the nucleus accumbens into dopamine release was decreased. So based on this result, the cocaine psychomotor response was decreased after the acupuncture stimulation. I really appreciate your listen. Thank you for your attention. Thank you.